The next talk is about a very, very important topic relevant for underground hydrogen storage, which is about microbiological aspect of the storage. So here I have my good friend and colleague Nicole Dopffel from NORCE in Norway here. Nicole studied biology at the university in Tübingen in Germany, where she received her diploma on microbiology. Actually, she has written some technical terms here that I can't really say about those microbes and what a special aspect of microbiology she has studied, so please forgive me for that introduction. From 2014 to 19, she worked as lab team leader and research scientist at the R&D department of the chemical company BASF in Germany, where she mainly worked in oil field related topics and microbial contamination problems in the industry sector. So she's a reservoir microbiologist. In 2019, she moved to the Norwegian Research Center, NORCE, in Bergen, Norway, focusing on different aspects of microbial effects in the energy sector. She also holds a yearly webinar on microbiological aspect of underground hydrogen storage. So follow her in LinkedIn or other posts that she will have, so that she has always the webinars and allowing you always connect online and learn about microbiological aspect of underground hydrogen storage. She's also co-leading a subtask on this subject in the IEA task force of underground hydrogen storage that we have presented yesterday. Nicole, we are all looking forward to learn more about microbiology.
Thank you. Super. Okay. Then thank you very much for the invitation. I'm very happy that I can present this topic here to you. For all the online participants, you can already open the website I have shown, menti.com, because we will have like a sort of quiz for you to interact with. And me as well, you here can also take part, of course. Okay, so I like to start my... What's Menti? That's a website. Yeah, what's Menti? Menti.com, it's for like gathering, like for quizzes, and you will see how it works.
I got an explanation.
Okay. Yeah, harmless, exactly. So first I want to start, like, why are we talking about microbiology when we're at the summer school for hydrogen storage? So I would like to ask first, like, where do we find microbes? And the easy answer is almost everywhere. So you can see some examples here. You can find them, of course, in water, soil, in rock, on our skin, in our guts, in the guts of animals, in hot springs. You see a beautiful picture of the Yellowstone Park here. And so, of course, also reservoirs. When I talk about microbes now in the rest of my talk, I will refer to two types. You can see here. Oh, it's not, the animation is wrong. Okay, but so very important, two types. So the first one is bacteria. You probably heard of that. The second one is the archaea, which are not so well known. The third branch of life is the eukaryotes. That's where we belong. I will not go into detail there. So when we talk about microbes in the subsurface, the bacteria and the archaea are ones to consider. So they look very similar. Mostly unicellular organisms, similar shapes, similar sizes, but they have very different differences. Also, we can spend a whole lecture on that. We will not do that. So they have differences in the cell membrane, cell walls, and also in enzymes. And for you, very important is for bacteria, they can make spores. Spores are ways of survival. Archaea cannot do that. Bacteria can have typical shapes of spheres, rods, filaments. And archaea can actually have all the shapes. You can see here a picture of a very beautiful archaea, which has the very fitting name of Haloquadratum. It was isolated in Egypt. So this is actually an archaea, which is square. So if you see that under the microscope, you can say, it's an archaea. That's already sure. And also important, if bacteria see CO2 and hydrogen, some of them can form acetate. And for archaea, if they see CO2 and hydrogen, they can form methane. There's a big difference. So you will not find bacteria who produce methane. You will not find archaea who produce acetate. At least no one found them yet. As a very rough rule, you can say archaea like to be more under extreme environments, hotter temperatures, higher salinities, for example. Bacteria like it more ambient, but it's just really, there are always exceptions. But then what is actually ruling or governing the community shape? So there are several factors which are super important, and the most important factor is for sure temperature. So there has been life detected from minus 20 degrees to 122, as long as water is liquid. These are now the limits we have. So if your reservoir is over 122, you can be sure there will be no microbes. But here I always say, if you have a reservoir which had 130 degrees in the beginning and you operated for decades, even making a water flood, there will be cooling effects, so your reservoir will not be 122 anymore. So please always consider cooling effects near the wellbore region, and then suddenly there's actually temperature where life can exist. So please always be careful with that. Then pH, an important factor. Most microbes can live at neutral pH, pH 7, but there are specialists going from the whole range from 0 to 12. Salinity, no problem. Up to saturation, you will find microbial life dealing with that. Metal concentration, the same. It is a stress for many of them, but they are specialists again. Pressure, there has not been yet a limit for pressure identified for life. So they can deal all the pressures we tested. Important here is the pressure difference can't be too fast. Like if you release the pressure very quickly, it will just disrupt the cells. But so you always go slowly. They actually have a very, very high pressure limit of who knows. Permeability is another factor important for the subsurface. If you have really, really tight reservoirs going into the millidarcy scale, there will be just no space for the microbes to live. So this is an effect of maybe for carbonate reservoirs. If it's really, really tight, then they could be sterile because of that. And then available nutrients. So for the permeability, you can maybe ask, okay, what is the typical size of a microbe? And here, again, really roughly, you can remember one bacterial cell has a size of 1 micrometer, just as a ballpark. But there are exceptions, a lot of them. There are many, many small, much smaller and much, much bigger. And then the question, but how big? So here we come to our first quiz. And now the online participants, but also you, can take either your cell phone and punch in this number. And I will now go, you will see it again. And hopefully this now works. If it does, it will be fun. So one single cell, your estimate, I will not give you multiple choice, that's boring, just an educated guess. What do you think is the biggest cell? So somebody wrote, yeah, something like this, 3 millimeter. That's what I want to hear, a number with a unit. So of course there are chains of cells which can be very long, but I'm talking about the biggest yet discovered microbial cell. I can tell you the second biggest one was discovered in 2002, which is called Theomargarita nabiensis, isolated in Namibia, which has the size of 750 micrometer. So what some people want to shout, you can also just shout. So what do we see? Okay. 1 centimeter is, for now, the one which the most answers. Let's give it a few more seconds. Well, it works. That's nice. 1 millimeter is now the most answers. So the bigger the number, the more people have actually answered that. Okay. So most people actually think a millimeter, maybe a centimeter. Okay, now let's see what it actually is. So it was actually recently discovered, published this year in Science. You can see a beautiful picture. It's actually 2 centimeters, one single cell. So it's fascinating questions now arising. How do they manage all their metabolism, DNA replication, which is actually based on diffusion, like everything? How can they deal with that? Big unknowns. It has the beautiful name of Margarita magnifica. So here you can see there's a lot of microbial diversity still to discover. Okay, so they have been isolated from mangroves, from a mangrove forest, I think in French Guinea. So what are actually now microbes living on? So metabolic activity of the microbes, it's what we are interested in. So this is now a simplified overview. We will start that we have organic matter input into our sediment, soil, or whatever. So you will have degradation of your organic matter into smaller ones, sugars, lipids, amino acids. Then you can have fermentation, also by microbes, degrading this further down into gases or small organic acids like acetate. And then there comes actually the so-called redox cascade. So that means that first there will be microbes, if there's oxygen, they will use oxygen as the electron acceptor, degrading these electron donors and organics. This is the oxic zone, this is where we live. But then the oxygen will be depleted very fast and you come into the anoxic zone, which is, of course, the most relevant for the subsurface. And then first you can have nitrate reduction using nitrate. Then there can be manganese reduction, iron reduction, and at the end there can be sulfide reduction and methanogenesis. They all have certain products, you can see. And this redox cascade also means that they are in direct competition. So if you have, for example, nitrate available, nitrate reduction gives much more energy to bacteria or to microorganisms compared to sulfide reduction. So nitrate reducers will outcompete sulfide reducers. This is also the reason why you maybe have heard that the oil and gas industry sometimes injects nitrate into the oil fields to fight sulfide reduction. That's the reason, to outcompete, so to go up in the redox cascade. Of course, this is now super simplified. You can spend lifetimes and lifetimes, and people do, just studying one of these microbial cycles. I will show you now the very interesting microbial sulfur cycle, as when you can call it. I will not go into detail. I just want to flash you with the complexity of everything. So there are multiple steps microbes can take back and forth. This is just now the biotic ones. If you now also consider abiotic reactions going on, this gets even more complicated. So there's a huge complexity already just in the sulfur cycle, going from sulfide to H2S and back. But the H2S is, of course, something we are all very familiar with. Sulfide reduction is a big threat, also for hydrogen underground storage. And we all want to avoid H2S. If you wonder, H2S is this really annoying smell of rotten eggs. You probably all know it, or like sewer smell. It's like this really strong thing we all react to because it is very toxic. So our nose is actually quite sensitive to detect H2S. But then again, what do you think is the lower detection limit of our nose for H2S? So it's the same code. So let's go to the next one. So what do you think is, where can our nose detect H2S already? Just, again, a number with a unit would be nice. Okay. So let's see, we have 1 ppm, 10 ppm. So I can already tell you, so as long as you can smell H2S, that's good. Because it gets really, really toxic when you can't smell it anymore. Then it's actually, then it's actually one, like when you go up to maybe 1,000 ppm, 1 H2S breath and you're on the floor. So it's quite dangerous. So that's also the reason why our nose is very, very sensitive. So the most answers we have is 1 ppm. but I also see 300 ppm. Yeah. Yeah, okay, there's a single one. Yeah, 1 ppm is the most answered. Okay, let's see what actually the answer is. You will be surprised because our nose is actually a very, very sensitive GC. We can smell it with 0.47 ppb. So this is actually where people publish with like, our GC can measure this. Wow, ppb. This is actually what our nose can do. And this is actually already showing how important it is for us. When we start smelling it, we run. And actually the first health effects come from 10 ppm, and people describe 10 ppm, it's so intense, you can't stand it. You will not stand in a place where you smell 10 ppm H2S. This is just done by nature to protect us from this toxic gas. So you see, it is actually very important to consider this. But we're talking about hydrogen. Back to this, and then again, hydrogen, methane, okay, why? We're storing natural gas since decades and decades. There has never been a problem. This is true, but natural gas is not hydrogen. We've seen this for thermodynamics, wettability, we talk about the differences. It's a huge difference for microbes. Hydrogen is a very, very, very good electron donor for microbes, for many of them. It has been a universal electron donor for thousands, millions of years. So you can actually see here now all the reactions, how they can fuel the different microbial metabolisms. And hydrogen can fuel all of them. The enzymes for hydrogen oxidation, you can find them everywhere. So that means a lot of different metabolisms can be fueled by hydrogen. There can be actually, again, competition going on if the nitrate reducers will want to out-compete the sulfate reducers for the hydrogen and so on, but there can actually be also multiple processes going on. But then you already guess probably the problem we have with underground storage. If you have microbes and you have hydrogen in direct contact, what will happen if there's food? So, and if you, I hope that you at the end will just remember this slide. This is the most important one. So microbes can consume hydrogen and they will have different effects. They can produce H2S, we talked about it. That means not only you will lose your precious hydrogen, but you will get instead H2S out of your borehole. Perfect, that's the worst case scenario. You can also have methane production, you can have acetate production. That only means hydrogen loss. They can react with a lot of minerals, they can trigger biogeochemical reactions, they can change the pH. They actually have an influence on all of them. Microbes can change the wettability, microbes can change IFT, microbes can change contact angle, that has all been shown. They actually can cause corrosion. It is another big topic, so microbes can actually eat steel, iron from the steel and corrode it very, very fast. And if you have this microbial growth in a porous space, and you can already imagine can plug. Microbial plugging is a big problem for porous media. So how this now looks, you can see here, this is within data now for me, which I obtained during the HYPE project. This is a project NORCE is doing with the University of Bergen, where we look at different metabolic groups of microbes and their potential hydrogen consumption. This is now a sulfide reducer called O-Lidesulfovibrio alaskensis. He was actually isolated from an oil pipeline. And you can see here has either 10% hydrogen in the headspace or 90% hydrogen in the headspace. And you can see over time how the hydrogen just declines. So he eats up the hydrogen. You can also see the rates, just as an estimate. You can already see the rates are different if you have different concentrations. And yeah, this picture here is just one of my bottles. If you want to do these experiments, I strongly advise to always put your bottles on the head. Hydrogen will diffuse out of your bottles. It's very annoying. It doesn't matter what kind of rubber stopper you will use, it will just diffuse out. So just a cheap way to prevent that, turn your bottles upside down. So when we now go into the hydrogen underground storage, so we talked about the different types. Gas reservoirs, salt caverns, and deep aquifers, and we now consider what we know about microbiology, you can actually look at this very differently. So with the gas reservoirs, if you are in a range where temperature, especially the temperature, is suitable, you will find microbes, probably always different for each reservoir. So there is knowledge that there will be microbial activity. For a salt cavern, it is more extreme, it's high salinity, but still there are, as I told you, salinity is not something microbes can tolerate. And deep aquifers, that's actually, for microbial standpoint, worst case. Aquifers are known for a super diverse microbial community. They will the highest risk of hydrogen consumption, for sure. So now I want to go into detail for the salt caverns and the porous media. So first start with the salt caverns. We heard about it already a lot. So what is important here? These salt caverns are leached very often with seawater. Seawater contains a lot of microbes, so we will actually introduce microbial diversity into the subsurface. And also, seawater contains a lot of sulfate. Additionally to maybe sulfate coming from some minerals like anhydrite. So you have a lot of salts, a lot of sulfate. So already not two things which are not good. And then also you have expected higher hydrogen input because you have this, the cycles will be faster, maybe some weeks. So the microbes which live at the cell walls where there will be water in the sump, they will see fresh hydrogen all the time. That means they can grow, they can build up cell mass. So the activity will be there, and probably the highest at the interface. And with the temperature and pressure in salt caverns, it's well within the range. So the temperature is not even that high. So we actually face a lot of problems within the salt caverns. And then you say, salt caverns, but it is so salty. Now the research is going on very quickly. So this is data from Stefan Janssen and Jan Gerritze from Deltares. They sampled a salt cavern here in the Netherlands. You can see pictures up there. And they incubated it now with hydrogen under 50 bar at 35 degrees. And you already can see a pressure drop during the incubation. And you know hydrogen has a lot of pressure. So if microbes consume hydrogen, you will initially observe a pressure drop. So this is already a sign there could be microbial activity consuming the hydrogen. This, of course, needs to be verified. There could be one of these annoying leakages again, but first data here. And there has been a very interesting paper published recently where they actually looked at the DNA based community. So DNA is something all the cells contain. We also, if we sequence them, we can identify. So each color represents a different microbial group. And you already can see that in all the salt caverns they sampled, also just the ones for natural gas, you will find microbes, pretty high actually, with 10 to the power of 6 cells per milliliter. And you find a huge diversity. So salt caverns are not sterile. You can see them, and you will find sulfate reducers in them. So it could be actually that over time, using the salt caverns, you will enrich for a community eating your hydrogen, producing H2S. So this is something to investigate further. There's still a very limited data on this. Also a problem because it's difficult to get samples from these salt caverns. But now with more interest in this topic, it's now coming with more publications and more knowledge. Then the second one is porous media storage. So we talked about this in the earlier talks. As I mentioned, it will contain a very diverse community, either introduced by the operation or just indigenous. They differ in every reservoir. And here the storage cycles will be longer. That gives the microbes actually more time to consume hydrogen. and which is very important, the large surface area in a porous media is, of course, there. That means there's the interface is much, much bigger. The microbes will always sit at the interface, so the consumption will probably more rapid. Then again, reservoirs, they vary very much, so always keep an eye on the temperature and pressure and salinity. And then here with the porous media, there comes the plugging into play. So again, some data on this topic. This is now from the BGR in Germany, from Anja Dormann and Martin Krüger. They sampled in their H2React project reservoir and incubated it with hydrogen, and you can see how the microbes consumed it and they refeed it with hydrogen, and then consumption again, refeed, consumption, and so on and so on. So this is actually, if you ask like a microbiologist, is this surprising? No, it's not. So you wouldn't amaze anyone at a microbiology conference with that. But I think it's very important for non-microbiologists to see that this is actually very easy triggered and very fast. Then also within our HYPE project done by my amazing colleague Na, we now did microfluidic experiments. So we have our microchip where we inject the hydrogen together with the microbes. This is now a halophilic strain. And you can actually see, this is now the different days, you can actually see the hydrogen are the brighter colors, how the hydrogen bubbles get smaller and smaller and smaller. You can see that the bacteria, you can't see it on these pictures, but we saw the bacteria on the interface between the brine and the gas, sitting and wiggling, and they consume, so the bubbles shrink together and shrink together. And that's pretty fast also with 7 days. So I think these kind of experiments, they're super important, and I hope we can extend on them and also visualize the microbes and so on, because this has, of course, a huge influence on everything on hydrogen underground storage. And also, what I want to mention here, it's not written, but we saw also a change in contact angle by the microbes. And then another example is by Diana Machado de Sousa and also Bart Lohmann's group from the highest free and H2React. Yeah, he's there. It was very nice to give me those data. So they are actually sampling now a lot of reservoirs and salt caverns, incubating them again in bottles and also under high pressure, which is always very cool. And here just some pictures of the sampling and the first results. And you already can see, so they have 2 salt caverns, 4 porous reservoirs, different temperatures, and you all can see that there's quite a lot of activity going on. Also in salt cavern A. Salt cavern B hasn't shown anything after 2 months, but then again, 2 months is not that long. From a different project conducted in Germany, they have sometimes, until the microbes really get started, it can take 300 days, lag phase, and then if they really switch on, they multiply, and then it goes faster and faster and faster. Yeah. Yeah, so they will multiply. So the problem will increase over time. So this is already showing, this is not something which is maybe a problem for a certain reservoir here and maybe a salt cavern there. It is a big problem over the whole range of reservoirs and salt caverns, and it hasn't been looked at very intensively yet. So that's also the reason why really it's important to look at the microbiology, because if you now choose your reservoir, for example, on all the knowledge you have on, not that deep as we heard, maybe the shallow ones are better, but the shallow ones are not that hot. Then you have more microbial diversity, maybe more cells, and then your microbial consumption may be much too high to be used as a storage. But then again, how are the rates? Are we really, maybe is the consumption very slow, maybe a few percent? Nobody knows. Maybe it will go faster over some years, but then again, we want to use those storage sites for decades to come. So a lot of interesting questions. And here I want to also mention the Underground Sun Project. We heard a lot of that yesterday, which was actually showing the first time also the microbial activity in the field. It's very, very important always to see the lab results not always correspond to field results. We all know that. It's for microbiology the same problem. So at the end, a field test always needs to show what is up. But here you can see the lab results, you can find it in the end report. You can see hydrogen is consumed, methane is going up. So actually there was archaea producing methane in this reservoir. And it was also shown by the field trial where you can actually see they did not recover all the hydrogen because it was converted by microbes, which then gave rise to the new idea of using actually a reservoir as this methane producing bioreactor. So that was the reason. And it's the same with the Haichiko reservoir. Yeah, so many open research questions which still need to be answered in the upcoming years. So which microbial types will be crucial for underground storage? Will it be the sulfide reducers? Will it be the methanogens? Maybe something completely different. Is it always the same family? Could be also, it's more basic for the microbiologists, but it could be always the same members, maybe. Maybe we can look out for certain types and say, these are really strong hydrogen consumers, don't use this reservoir. Will it be always the same, especially interesting for the salt caverns? Or is like every salt cavern like a small own universe where you always find different ones? So that would mean that you would need to test each single cavern for the microbial activity. What are the microbial rates? This is, of course, always important for modeling. You may ask, can I now model the microbial hydrogen consumption rates? My answer, I don't know. Probably, but you always have to be sure to use correct data. So we are now actually producing the experimental data. So please don't just go into literature and just pick up some rates here and there. Of course, you will find a lot, but this will be not valid. So there will be always differences with the community, the cell number, how much hydrogen there will be. So here I want to actually quote Bart Lohmans on this modeling thing when you say, crab in, crab out. And I really like this quote. I think it's very fitting. So always use experimental data specific for a reservoir if you can. Also, like, it would be similar if I would ask you, please model the feeding habits of the Dutch population. Or not even, that's just one culture. Model the feeding habits of the people of the whole northern hemisphere. And I am sure there will be very smart people who can come up with a model. But if this is then actually depicting my feeding habits or maybe the one of my baby, it could be different. So always consider they are living beings and they tend to act differently.
Are they stubborn as well?
They can be stubborn. Every microbiologist has like their home stories, like I had a microbial strain during my PhD and it's just stopped growing during the summer. Like 6 weeks, it just didn't grow, nobody knows. Probably. Exactly, vacation. I tested all my frozen microbes, my stocks, everything, and after 6 weeks, it just like this started again. Maybe the moon, the weather, pressure, who knows? So it's really, they can be really like little idiots, really. So please consider this in your models. It's not just a particle, it's not just a reaction. So that's also making modeling of microbes super difficult.
You visit psychologists?
No. No. But you see, I still remember, so it stays with you. Then pressure effects on microbes, a topic which has not been investigated a lot, but very fascinating. I think there should be more, especially with the hydrogen. It's interesting because the higher you go, the more hydrogen you get dissolved, and the more hydrogen dissolved, the higher the microbial rates will be. But then again, these huge amounts of hydrogen in the subsurface are completely artificial. You will not find that in the environment. Microbes are used to nanomolar hydrogen normally. So it can be actually, if you put a lot of hydrogen on them with a high pressure, that they actually can't deal with it. So there could be actually certain thresholds to identify. So maybe we say we need this amount of pressure and this amount of hydrogen, and it will be, the activity will be just choked. Could be. Yeah, that's what I said with the thresholds. Maybe salinity comes also in. Then inhibition, always a question, can we inhibit them? Of course, there are biocides. The oil and gas industry can talk a lot about this. And always be aware that if you already have the problem, if your reservoir has started to grow, you have biofilms and activity, it's super, super difficult to control them. So it's always better to act first, to think about monitoring, how do you actually follow this, before you just wait and then it's too late. So treating a reservoir is very, very difficult. Then the trigger geochemical effects, also a point. They will interact with minerals, they can dissolve them, they can precipitate them, pH effects and so on. But then again, maybe they're also usable as a leakage detection method. So if you actually have hydrogen leaking out of your reservoir, and you always find certain types growing, maybe you can use them to pinpoint leaks on the surface. This is actually coming from idea also from CO2 storage, where they're already trying that. So could be, it's pretty far, but could be an interesting topic. Yeah, and with this, I want actually to end. And while we talk about questions, the online people can use a different code here and just put in, so what do you think now from your scientific background, what is the most urgent question you would now say this needs to be answered for underground storage, considering microbiology? And I just need to close this, because it's I don't have the paid version of this. That's the reason I will have to go here. So here's the code, and we now can so the online participants can write in. What do you think? You have multiple answers. Don't make it too long, just single sentences. And then I want to thank you, and I'm open for questions.
Thanks a lot. Thanks a lot. Fascinating talk. Plenty of questions online and off here as well. If you don't mind, I go this way. Or ladies first. I go first to you. Of course.
Thank you very much. It was a great presentation. Well structured, message delivered clearly. Well done. I'm Lubika from University of Edinburgh. And I've got a question regarding the recovery of the injected hydrogen. If we look at the simulations that have been done so far, so we can say that after the first cycle, you've got the highest losses. And then when the system stabilized, so efficiency factor for the next cycles, further cycles, is better. However, I suppose that those simulations didn't include any microbiology. And as you've just presented, how we go through the further cycles, they've got freshly nutritions, and they can just increase the colonies and
the cell
population.
Yeah.
So if we couple those two, do you think that improved recovery factor for the further cycles would not look like as we can see them now?
Yeah, exactly. So you can actually expect with more cycles, more time, that means more adaptation. So the specialists will come, there will be higher cell numbers, and then the consumption will be faster. So it can actually counteract that you will lose more hydrogen. It will cancel maybe each out. Yeah. Yeah, exactly.
Right, let me start by saying congratulations for a fantastic performance, brio and enthusiastic, so I enjoyed it. Question. A lot of energy companies at the moment, they're looking on salt caverns to repurpose them.
Yeah.
Existing caverns, which had already methane inside for years, they're going to be repurposed for hydrogen storage. So my first part of the question is, in the natural gas storage caverns, do we have microbial activity? We do.
Yeah.
So this type of microbes, will they survive, or there'll be a completely different family that will start as soon as hydrogen throws in, begins to be thrown into the cavern?
Yeah, that's a really good question. So that is also something a lot of microbiologists are now interested in. So what happens if they see hydrogen for the first time in this environment? So what will happen? It's not clear. There will for sure be a community adaptation going on. Then again, how fast that will be, I don't know, probably relatively slow, because the cell numbers in the salt caverns start slow. But as far as I know, especially in Germany, there has been now quite a lot of sampling going on. Each salt cavern they looked at, even if it has been used for oil or natural gas for decades, they always found microbes. They never found a sterile salt cavern. So it will be a problem for all of them. The question is still, how big is the problem and how fast will it come?
But so far, the microbes that we had in the salt caverns that are used for natural gas, maybe we'll have, and I'm sure, accept your argument, we'll have microbes, but we have no problems.
Yeah, then again, natural gas is not hydrogen.
Yeah.
Microbes can't grow. I mean, there are specific microbes which can use methane, but not in the salt caverns. Hydrogen is a universal electron donor. It's completely different from methane. So if you say methane was not a problem, it's clear, because the microbes can't use it. Hydrogen they can use. So it's a completely different thinking. It's like with the materials, methane, hydrogen. It's similar. So sure gases, but it's different. Yeah.
Thank you.
Yeah.
Thank you, Chris Banks, Schlumberger. What you've just presented to me is an entire presentation on project risk, which scares the hell out of me. So going back to salt caverns, so I understand biocide in the oil industry when you're injecting it into a tight pore space. When it comes to a very large void space, what remediation efforts is it possible to take to make sure that we aren't ruining the economic resource we're putting down there?
Yeah, that's a question I get a lot. And of course, I think with a salt cavern, the chance is to get it under control, because it is a confined space. There's not a lot of input from fresh cells or something. It's quite high. I think we can control it quite easily. There has not been, at least not published, any inhibition studies. I mean, this high salinity is quite tough for biocides. So there's probably the biocide vendors need to think differently how they would do that. But I think it will be controllable, maybe also by changing the pH or by blocking the interface. I think the salt cavern has good chances to actually get control, but it needs to be studied first.
Until the next question arrives, because I was lazy to go up. So is there any treatment so that we can, let's say, inject, antibiotics in the reservoir?
Yeah, never call it antibiotics.
Very creative.
Call it a biocide.
Yeah, alter the biology.
Yeah, if you say inject an antibiotic, like regulators.
I guess government folks left yesterday. So that's,
yeah,
Yeah. Taking notes? No.
Yeah, so like I said, like the biocide choice will be a focus, and it will be interesting. For the salt caverns, I think it will work, but again, needs to be investigated. Until now, we don't even know what we're dealing with.
Great presentation, Nicole. Hamid Nick from DTU. You mentioned about how important it is to work with real data from the lab.
Yeah.
And I'm sure you agree that we also need to have a real data from the field. We are going to basically store CO2. And the communities vary from one field to another field. What sort of your method for sampling? Because we have one well in one location, and we may sample at a specific time in the reservoir at a certain condition. we get some sort of a idea of what is the microorganism living at that condition, at that location. That's not representative of the whole reservoir, whereas when we inject hydrogen, hydrogen will be basically active in the whole reservoir and may see microorganisms that way. What would be your recommendation to basically characterize the microbial community and how they would evolve also during the project life?
Yeah, very good question. Of course, it's always advisable to use original microbial communities. So always use the field fluids, not just standard strains. And then sampling is a whole topic. I think we have been on workshops and conferences where we've just sessions about how to sample this correctly. It's not easy. Of course, especially if it's offshore, you can't go there, and sometimes they just fill it into a Coke bottle and give it to you, and it's like, So it needs to be, of course, sterile. It needs to be kept oxygen-free. It needs to be transported fast. Then again, if you take fluids on the wellhead, is this representative for the subsurface? Questionable. But then, of course, the best way would be subsurface sampling. But then again, this is costly. It needs a workover, stop operation. So finding an operator who would be willing to do subsurface sampling, it's very slow, low. So sampling, it needs to be thought through. But then again, we always, we need to work with the operators, what is possible. So we always need to find the best way in between. So keep it sterile, keep it anoxic. If you have to choose between sterile and anoxic, keep it anoxic. And then it's always just an estimate. At the end, as I said, the field trial needs to prove. It's always the case. But then again, I all invite you to, with all your experiments on wettability, core floods, repeat your experiments, this time with microbes. Write me an email, I will send you some, and then let's see how different the results will be.
Don't send us the stubborn ones.
The easy ones. Bart.
Hi, Nicole, thanks a lot for the presentation. Nice presentation. I don't have a question, but if you allow me, I have some comments too.
Sure.
So many people think like, okay, also the question, they might not be very different. They might be very different in one place in the reservoir from the other. That's fully true. Now, I guess some people will visit the site inside Wenning. I've been there for multiple sampling campaigns. I'm not criticizing the guys. I know that this is simply how it works, right? But if you see from microbial perspective, you will see at such a site that it's absolutely not a contained system. So things go in, things go out, things go back in. And these are all what we call in microbiology inoculation events. So as a microbiologist, you would look like not necessarily only what is there in the subsurface, which is already there, but also which might be introduced. But much more, if there is a niche, I guess most people know the name of or the definition of niche. If there's a certain condition that they can be active, it will be. Yeah. So that's why we usually look at these things more than what is necessary there from the beginning, because that can change completely. And one comment on the hydrogen. Sometimes it's good to look at the natural equivalents, right? We don't have hydrogen reservoirs, and that has a very good reason also. In many cases, there is methane from abiotic source, true, but there's a lot of biotic methane, and that's the reason. It's a much more stable molecule. If you talk about hydrogen, completely different thing. Right? If you talk about reservoir souring, oil versus hydrogen, different worlds entirely.
Exactly. Yeah.
Thank you. Let's thank Nicole once more for this excellent talk.
Thank you.
Nicole.